My presentation is a halocyte as a nanotorpedo for enhanced drug delivery through cell membranes, brain barriers, and skin. My name is Saleh, and my um, co-colleagues are Prajapati, Rahman, Anna Stavitskaya, Dr. DeCoster, and Dr. Lvov. So first we have some SEM and TEM images of halocyte and its penetration through into the cells fluorescently, which is this image on the right here with the blue fluorescence. We also have TM images on the right here demonstrating gold loading into the tube's lumens. In the upper right corner is a buildup of halocyte and what our goals are. We also have two papers that we published recently on delivery into endothelial cells and synthetic blood-brain barrier membrane. So
Maddie,
halocyte.
I'm sorry to interrupt you. Could you switch over to presenter mode, please?
Oh, okay.
Thank you.
Let me see if this works. Okay, does that work perfect?
Yes, that's fine. Thank you.
Okay, thank you. So halocyte can be utilized for many different objectives such as drug delivery, tissue engineering, wound healing, and hydrogels. This is because halocyte are natural biocompatible aluminosilicate tubules, which are usually derived from volcanic dirt. The tubes are oppositely charged on the outer shell and inner lumen. The inner hollow lumen is positively charged and the external surface is negatively charged. We have a couple of SEM and TEM images here to show what they look like structurally, as well as zeta potential, which shows the surface charge. So we can see that the surface is negative as well. And we also have a picture here of halocyte in what you would see in the naked eye. Okay. Oh, and the structural dimensions of halocyte, they're roughly 50 to 60 nanometers in external diameter with an internal diameter of 12 to 15 nanometers and a length of 500 to 800 nanometers. Okay, so halocyte properties and how we can selectively load different types of compounds such as drugs, flame retardants, polymers, enzymes, and proteins. It's dependent on pH charge and how soluble it is in water. The difference in cationic and anionic surfactants when loading and their dispersibility is actually quite large. We can see that it shows a surface charge of -30 with a dispersibility of under sub 2 hours, where when we have a anionic surfactant, it's up to -65 millivolts with a dispersibility greater than 40 hours. So it can be dispersed over multiple days. Okay, and then another method of additionally through the initial like pH and other properties, we can selectively etch the inner alumina where we increase the loading capacity of the halocyte from 15 to 30%. This is usually done by stirring halocyte for roughly 8 hours in 1 m of sulfuric acid at 80 degrees Celsius. The point of this is to remove only a small amount of the inner lumen to increase drug loading capacity without completely destroying the structural integrity of the tubule. The graph here displays the lumen diameter as the acid etches the inner lumen away. So we can see the diameter increasing while the inner lumen is also decreasing in structural integrity, but not fully to where it's not able to hold any compound any longer. So this is what typical data when analyzed will look like when using halocyte for sustained drug release. Here's some examples of us using compounds such as dexamethasone, furosemide, and nifedipine. These graphs have usually the same trend. It starts with the initial 20 to 30 burst release of the compound, then slowly over many hours and days, continuing the release of whichever drug you're using. So we can see that initially the burst is somewhat large, not a lot, and then it stables off and continually goes over a long period of time. It could be 10 to 24 hours or multiple days, depending on how the formulation was constructed. Okay, and this is a short animated clip to give you a visualization of how the initial burst occurs, and over long term, you see that the compound is actually attached to the inner walls because of the electrochemical charges, and how that's usually why it slowly releases over long periods of time. Now that I have gone over the basics of Halocyte and drug delivery and the different characteristics of the tubules, our goal is to load these nanotubes with drugs and pass them into cells or through more complicated bodily membranes and even skin, exploiting the idea of the nanotorpedo, which is partially perpendicular penetration. First, we have an enzyme-activated intracellular drug delivery with tubule clay nanoformulation into cells, which was demonstrated by Dr. Fakhrulin. So what exactly was done? Well, brilliant green dye was selected as a simple anti-cancer model drug for loading into the halocyte lumens. After loading was complete, dextrin stoppers were placed at each end of the tubules to prevent leakage of the dye. Then was delivered to the cell where the stoppers degrade due to biodegradation properties, and the dye releases into the cells. So one thing I do want to say is that Dr. Lvov was the first to show how halocyte could enter the cells, but Dr. Fakhrulin was the first to display that with drugs loaded inside the halocyte. Here we can see TEM image of the brilliant green loaded inside the halocyte, as well as the dextrin stoppers being placed, and the difference on a open lumen versus the dextrin stopper being placed on the outer edges of the tube. And we also have an AFM, which is done to confirm the fabrication of the dextrin caps to show that is in fact plugging the halocyte openings and stopping the release of the initial burst of the dye. Okay, so here we can see the toxicity of drug-loaded nanocontainers towards A549 and Hep3B cells, which are two different types of cancer cells. These are resistant assay results demonstrating the LD50 value of BG-loaded HNTs for A549 and Hep3B cells. The twofold increase of LD50 and Hep3B cells occurred due to the reduced uptake of halocytes if compared with the A549 cells. So basically, we see that when the halocyte actually enters the cells, the percentage of cells that live is increased by a lot over just having halocyte with brilliant green without the dextrin stoppers inside, or without the halocyte actually going inside the cells. Because A549 formed crater-like concentric circular regions around the nucleus. In Hep3B, the nanotubes were detected mostly on the cellular membranes, contributing to the increased height of the cupola-like positions. These suggest that HNTs are preferentially internalized by both cells. HNT internalization has a higher proliferation of cells, which is a characteristic of malignant cells. So, like I said, you can see here on figure B, the percentage of cells that live is much greater with the dextrin caps on the halocyte that's loaded with brilliant green over just having halocytes with brilliant green even inside the cells. And these are some more images showing the actual dextrin capped halocytes in different cells after 24 hours of incubation. So we can see the difference that occurs along with the same results, showing how and why halocyte are advantageous over time rather than just instantly bursting release into cells. Okay, so halocyte nanotube penetration through the membranes. cell membranes, permeability. So our goal is to show the permeability of halocyte through the blood-brain barrier membrane consisting of two cell types, including interioration and passing through the skin. We will see if it works or not. And number three, skin multi-component barrier passing for an efficient dermatology drug delivery, which is currently in progress with us. That is some of our current work. In such penetration, there's higher probability to pass the membrane with perpendicular orientated nanotubes, which show them as an efficient cross section of 50 by 50 nanometers rather than 1 micrometer in a horizontal location towards the membrane, which is what we've noticed and what we predict. Okay, so our first attempt was Hnt penetration into the cells. So this is a schematic of the first experiment of our work. And we started by taking dissecting rat brains and taking the endothelial cells from them and placing them in culture flasks, which we then incubated and grew to our desired amount. And then we placed them in cell culture plates with media where we stimulated them with two different types of loaded halocyte. So we loaded one with RITC, which is a red type of dye, and we loaded them with ionomycin, which is a calcium ionophore, which is also used for anti-cancer treatments. And we had good results with both of them, and we analyzed the data, and I will be showing you that here in the next few slides. Here we can see first we did the Hnt, the halocyte and drug characterization to make sure that everything was working properly. So figure 1 displays the rate at which the dye is released over a period of 24 hours. Once again, this trend should be the same for most applications where there's an initial burst of roughly 25% within the 1 hour time frame, which is then is stable. It gets stable and tapers off over the next 24 hours over the same amount of release. And figure 2 is thermogravimetric analysis of the different compounds. So we have pristine halocyte, halocyte loaded with ionomycin, and halocyte loaded with rhodamine. This shows the amount, this can show the amount of drug loaded into your halocyte. We had roughly 5 to 8% weight loading, which is quite normal for both of our compounds. Okay, so RITC delivery into the endothelial cells. These are our initial results after adding the samples to the cell culture plates of endothelial cells, which make up the major part of the blood-brain barrier. Figure A and B show RITC alone, and then figure D and E display RITC that's loaded in halocyte. So we can see that the fluorescence is so much greater with halocyte because we believe the halocyte is binding and partially penetrating into the endothelial cells, causing them to release the dye inside the cell nucleus and make them so bright. And we predict this because there is no coloration on the outside of the cells. Most of the color is being generated inside the cells, which leads to us believing that it was successful in reaching the cells. And I'd like to mention that the halocyte we used were not modified in any type of way. It was simply mixed with the drug and loaded into the sample. So there was no external modification of the tube. Then with our ionomycin sample, which was the calcium ionophore that we utilized for drug delivery, is we had to use real-time calcium imaging to observe the results. And the initial response that we had, we did 3 different conditions to make sure that halocyte was the best. Is we did ionomycin by itself, and we did RITC by itself, and then we did HNT loaded with ionomycin. And these are on astrocyte endothelial cells. Figure A is ionomycin only, where with ionomycin, you have an increased spike, a huge spike of fluorescence, and then it tapers off. And then with figure B is halocyte alone, which showed only very dim fluorescence, but for a long period of time. And then when we combine them both, we got figure C, where it was a bright fluorescence color. The cells were responding really well to the calcium over long periods of time. All cells were stimulated with ionomycin at the end of each experiment, which was used to show that the cells were still responsive to calcium changes, which showed that there was no cell death. Because once the cells stopped responding after your initial experiment, that shows that they were dying. And when we tested it with calcium at the end of each experiment, it shows that they were still alive and responding really well to any sort of treatment. I would also like to mention in the previous picture here, we kept cells with these samples for 10 to 12 days, and they were still looking very normal and were not stressed at all or dying. So this shows that the halocyte is also non-toxic towards these blood-brain barrier cells. Okay, so this bar graph is data analysis of the previous images I just showed of the calcium imaging. And I showed many graphs here to show all the different conditions, but the main focus I would like to keep is on figure D. So these bar graphs are obtained by averaging the values over the region of interest from each condition used for the experiment. At least 5 conditions were tested here 3 times. Overall, the number of cells analyzed were 1,232 for all wells and conditions reported in the calcium imaging. So I know the picture might be a little blurry, but in figure A, we can see what I was talking about initially, where the initial spike of ionomycin alone occurs, and then it tapers off here, and it doesn't really go anywhere after that. Whereas in figure B, we have halocyte loaded with ionomycin, how it initially spikes, and then you see how it continuously tapers up and consistently goes at that level for a long period of time. And then figure D shows that why the cells aren't being stressed is because when we did the halocyte load with ionomycin sample, it increased and then stabilized and continuously went. And then we even added ionomycin alone after that, and it's went even higher and continuously stabilized for a longer period of time. So the cells respond really well to halocyte. They have absolutely no issue or toxicity problems with halocyte because of these results. Okay, now this this graph displays is a signal analysis scheme where B and D are parameters obtained from each calcium signal averaged over the region of interest, ranging from 41 to 140 for each condition in a single experiment, and then a range of average for 3 experiments. The error bars represent the standard deviations in between the experiments. So we can see that the cells responding to stimuli were much greater for halocyte ionomycin over the halocyte alone and the ionomycin alone. And the peak calcium response was the greatest with halocyte with ionomycin. We used 2 different concentrations of halocyte with ionomycin to determine if a greater concentration of halocyte was better or worse, because sometimes we can predict that too much halocyte might be not efficient as compared to only a small amount. So we did 10 nanograms and 50 nanograms, and the more we used, the better it got, the better the cells responded to it. So it was not what we expected, but we are happy with these results here. And then our most recent work, where not only did we use endothelial cells, but we constructed a synthetic bilayer of membrane on top of a porous membrane. So we took a transwell cell insert and that had a porous membrane on it, and we layered a layer of endothelial cells and astrocytes on top of each other to synthetically mimic a blood-brain barrier to help to give us more realistic results with our experiments. And so we took the transwell insert and we layered the two cells, and then we added our sample on top of the cells, and we saw how it diffused and if it diffused through the membranes and even through the pore. Okay. So here in figure, okay, here in figure 1, we have controls which show the Hnt RITC fluorescence intensity with no cell inserts, direct addition to the bottom well, and B, 20 nanograms per milliliter Hnt RITC added to the support. And then C is just a control membrane support with treated with media alone. So you can see the fluorescence intensity increases by a lot. That is because the halocyte is working with the membranes, and it's passing through over longer periods of time. So we see here that at first, in the first couple of hours, the intensity is low, and over a longer period of time, the intensity increases. Okay. So in figure, for example, figure 2A through B displays some decrease in fluorescence intensity at the 4-hour time point. The co-culture membrane absorbs some of the upcoming rhodamine, but further increasing passing enhances the signal growth beneath the model barrier. This significance is the display of Halloysite nanotubes being utilized as a drug delivery carrier efficiently, which we predict is due to predominantly vertical penetration through the cell membrane. And then here are some images displaying the multi-layer. These are images, these are quick stain images of model brain barrier of co-cultured endothelial astrocytes plated on the porous membrane that I described. These were taken by non-inverted microscopy of transwell assay with a layer of 428 astrocytes and around 400 endothelial cells. These are two Z-stack images with a 0.3 micrometer step size. So we see the two different types of cells here, and this is our initial goal. Which was very hard on its own, is to make sure that we were able to significantly show that we had two actual types of cell layers on top of each other. Otherwise, the experiment wouldn't work at all to mimic a blood-brain barrier membrane, because it is supposed to be one type of cell in front of the other or on top of the other. Okay, and our current work, and currently we're using a larger halocyte, and which has a length of 1.3 microns and a diameter of 90 nanometers for cosmetic applications for the skin in the form of UV protection creams. The halocyte in figure A and B are from Applied Minerals and Sigma Ultrich halocyte, which with an inner diameter about 12 to 15 nanometers, which is quite normal. That's the average halocyte. These halocytes we believe, are efficient for cell membrane penetration because they are smaller, but we have larger halocyte for skin application, which we believe will be better which we can see in figure C from North Star Minerals, will be more effective due to their inner diameter being around 20 to 22 nanometers, allowing a greater amount of drug to be loaded inside and have a better type of torpedo penetration into the skin. So here's some data from the larger halocyte that we recently got, and we see that the larger nanotubes from North Star displaying a diameter is around 80 nanometers and a length of 0.5 micrometers, which is larger and longer than most nanotubes on the market today. We predict these larger tubes will be advantageous in having greater concentrations of compounds loaded inside with a longer extended release time. Additionally, we feel like they would be better for skin applications because of the larger capacity and because the skin seems to be a lot easier to work with rather than cell membranes. So we can see the graphs here, which show the percentage and diameter of the cells, so an average usually, and it shows that it's much larger than normal your average halocyte. And here are some schematics showing our goal of our current work. So our current work is utilizing the nanotorpedo approach in products for skin applications. The figures display two possible outcomes that we are aiming to achieve. Figure A demonstrates a cream mixture that we will be making, which is lightly placed onto the skin. And we expect the halocytes to be in that type of, to be layered like that onto the skin. And then in figure B, we plan to deeply massage the cream with halocyte onto the skin to achieve a more perpendicular and in-depth application and to see the difference in both and to see which one is more effective or if either of them are effective. Current experiments are being done on stained pig skin. So that's the type of skin we're currently working with. And we're looking to start microscopy analysis as soon as possible. We haven't had the best results with microscopy analysis yet because of how specific the sample needs to be prepared and because of the type of equipment that's required, which we currently do not have access to analyze this data. In conclusion, we exploited Halloysite clay nanotubes for penetrative drug delivery, one through cell membranes, shown by Dr. Fakrulin's work and our own new results, the blood-brain barrier, and the skin. We developed a bilayer membrane of astrocyte plus endothelial cells as a model of the brain barrier and tested its Hnt permeability. We found that cell membrane is better permeable with smaller 50 nanometer diameter and 0.7 micrometer length halocyte and thicker barriers such as pigskin layers, more efficiently treated with larger 90 nanometer diameter and 1.3 micrometer length, allowing for a 20% increase in drug load. So the smaller halocyte, we see it's better for cell penetration and biological applications within the body, and then the larger halocyte better for outer things like wound healing of the skin and cosmetic skin applications such as like sunscreen. And lastly, we proposed a strategy on the clay nanotorpedo drug delivery through membrane exploiting and its perpendicular orientation having smaller 50 to 90 nanometers in diameter cross section. And acknowledgements. Dr. Fakrulin and Kazan University and Anna Stavitskaya of Gubin State Oil and Gas University of Moscow, Russia. And I would like to thank Dr. Lvov for being my advisor and my colleagues in my lab for helping me with all my work.
Thank you very much, Mahdi, for this excellent presentation. I wonder if there are any questions. Please ask.
If there's any questions, please ask. I'll try to answer as best as possible.
May I ask, even I am co-author, what is the reason, let's say you have small halocyte and big halocyte.
Mm-hmm.
Are the inner lumen the same? And if
No.
are the same, so larger tube have larger lumen?
Yes, larger tube has larger inner lumen and the smaller tube has a smaller inner lumen.
Which means that if we believe that they start rolling from larger diameter.
Yes.
It has to be discussed.
Yes. Yeah.
What do you think, Ravil?
That is an interesting and hitherto unknown question, as far as I understand.
So, as I understood,
needs to be discussed.
Applied mineral halocyte has lumen diameter, let's say 10, 15, and larger this other mine, North Star mine diameter is about 20, 21. So it's about 1.5 times larger. This is something has to be discussed and verified.
So we did the TM analysis of, we have many images of the larger halocyte, like here in figure C is an example, one of the examples. We have many images of two different types of larger halocyte, and the average of all of these samples we took, the average inner lumen was around 20 to 21 nanometers compared to normal halocyte, which is usually 12 to 15 nanometers in inner lumen diameter, not external diameter.
Okay.
Okay. Any other
We have
questions?
another question from Richard
Okay.
Brown in the chat. What is the ultimate fate of the Halloysite crystallites after they have penetrated the cell membrane, cell nucleus?
So our goal is to partially penetrate or use the halocyte eventually as even as implants for if we're talking about things like the brain as implants, not something that's going to permanently be there. But as far as skin goes, I think that it will be completely safe to use in sunscreen applications on top of the skin, but possibly not large amounts just inside the body for long periods of time.
Thank you.
One question.
Yes.
Why, sorry, Aranda, when you try to, or your goal to try to incorporate this clay in the cells, how do you propose, what is the driving force to force that the clay penetrate in the cells? Because I do not see how you will try, like, for instance, the nanoparticles or gold nanoparticles or iron oxide nanoparticles, they put some specific functions on the external surface of the particle to force or to drive the interaction with the cells. But I do not see how you intend to be that the clay go inside the nanocyte go inside of the.
So are you asking about these type of results here that I'm showing?
Yeah.
Okay, so we didn't go into this with any any actual. This is the first time this type of work is being done, so we don't know why exactly or how the halocyte is binding to the cells, but it seems to be electrochemically attracted because all we're doing is placing it. We're not forcing it, we're not driving it. It is by itself on top of the cell being attracted and going inside the cell. Maybe they're moving, they clump very fast, so they probably moving down and clumping and attaching and penetrating the nucleus by themselves. It doesn't seem to require a type of driving force to force them in. They seem to be attracted to the endothelial cells specifically because we have many results. This is just a fraction of it that shows where in every case scenario and at any time point, whether it's 30 minutes, 4 hours, 24 hours, 10 days, that halocyte loaded with the RITC had better fluorescence in imaging of the endothelial cells than RITC alone. If it was RITC alone in this image, the whole image would be red. All of the empty space would be completely red. You wouldn't be able to really see the cells. They would all be the same level of fluorescence. But with halocyte, the inside of the cell nucleus is brighter than outside or around. The inside is specifically brighter, very bright compared to any other results.
Yeah, but I wonder if you have the halocyte, and the halocyte with the fluorescent molecule is the one that go inside. It's not the whole. I mean, you are sure is the whole particle that go inside?
No, I'm not sure. No.
The halocyte release and the released molecule is the one with that is going inside. That's my point, because in other type of nanoparticles, you need to functionalize the particle that so to force the particle inside. So that's my point.
Yeah, we haven't done that, and we don't know if the halocyte is fully penetrating or partially penetrating or just binding to the cell and then releasing the rhodamine inside the cell and the cell is just absorbing the rhodamine. We assume that it's partially all three types. We don't know if it's just fully penetrating, partially penetrating, or binding, but we predict that it's all three types happening together. But we because not everything is the same. Halocyte aren't all the exact same shot size or the shape, or they don't all orientate the same way. It could be different angles that it hits the cell. It might be affecting if it penetrates or if it just binds and things like that. And we are trying to figure that out, and we're trying to get those answers through this work.
Okay, thank you very much, Magdi. And I think that with this we can finish this session.